What up, everyone? It's your boy Vape Memes 2020 back here with another video. Today, we're going to be doing something very different compared to my normal type of video. A while back, I made a video with my boy Beaumaths, and there we ranked biblically accurate angel depictions based on how biblically accurate those depictions actually were. And in that video, I said the following. I might do some videos discussing biology and theology, just because there might be... I have some fun things that might be interesting with that. Well, today is the day. Today, I'm going to be discussing how biology intersects with theology. And in particular, I'm going to be discussing how biology can actually be misrepresented for the sake of theology. Yep, that is right. Today, I'm going to be doing a debunking video talking about how the structure of DNA proves the existence of God. Now, as many of you could have probably already guessed just by the length of this video alone, this video is not just going to be a simple debunking of the claim that God has signed his name on our DNA. Instead, I want to do a lot more than that. I want to debunk that claim, but I also want to provide you with plenty of information about the actual structure of our DNA. Because by understanding the structure of DNA in more depth, you can then understand why the claim that God has signed his name onto our DNA is outright wrong. However, I do want to respect your time. So, if you aren't interested in learning about the structure of DNA and you just want to see me debunk the claim that God has signed his name onto our DNA, just go to the timestamp shown here in the top right corner of the screen. And that should get you straight to the portion in which I debunk the claim that God has signed his name onto our DNA. If you are interested in learning about the structure of DNA, however, then just sit back, relax, and enjoy yourself, and we'll get to that in a moment. But before I discuss the structure of DNA, I do want to give a brief preface to this video, because for those of you who are familiar with my channel, you might be wondering why I'm even making a video like this in the first place. Because I myself am a Christian. In fact, my entire channel is basically dedicated to both apologetics and theology in general. So this begs the question, why would I, as a Christian, go after another Christian who is providing an argument for the existence of God? An argument that could possibly lead people to Christ? Well, the reason I'm making this video is very simple. The claim that God has signed his name onto our DNA is just wrong. It's extremely wrong, in fact. There are a great many reasons to believe not only in the existence of God, but also in the truth of Christianity. I could probably give about 4 to 5 very well-supported arguments for the truth of Christianity just off the top of my head. The main issue is that the argument I'm going to be critiquing is a very far cry from the good arguments for God's existence. I believe that God deserves our best, and God himself even set this precedent in the Bible. For example, in the book of Malachi, God speaks to the people of Israel and chastises them for quote-unquote offering polluted food onto his altar. Because instead of offering the best of what they had as a sacrifice for God, the Israelites rather offered what was effectively their table scraps and unwanted animals as an offering to God. They were disrespecting God because they were offering to him sacrifices that were frankly inadequate. These were sacrifices that were so bad that most people wouldn't even offer them to other people, let alone the omnipotent, all-knowing, all-good God of the universe. And much like those ancient Israelites in the Old Testament, I believe that we today can also disrespect God indirectly by offering bad reasons to follow him. If we present arguments for the existence of God that are factually wrong, arguments that could be debunked with a bare-bones knowledge of biology, then I think it's safe to say we're not offering God our best. In fact, I would go so far as to say that doing this would qualify as possibly even taking the Lord's name in vain. Because it makes the idea of believing in God seem foolish, because you're offering such poor arguments to believe in God. In short, I am making this video because we as Christians serve a God who is, quote-unquote, the way, the truth, and the life. And if that is our God, we ought to serve him in both spirit and in truth. Of course, another reason I wanted to do this video is because it will give me an opportunity to help all of you learn about God's creation, and in particular, DNA. And truth be told, I'm really happy to be doing this, mainly because I actually did my major in biology when I went to university. So, if I have a chance to talk to you about all of that, well, I'm more than happy to take it. But with all of that out of the way, let's look at the video in question. It's quite short, so I'm just going to play the relevant section of that video for you, then we'll go and discuss the structure of DNA, and hopefully, in doing that, we should be able to better understand why this video is wrong.
In your genetic code, it's a double helix that right turns, and there are these sulfur bridges in the midst of every single atom that you have that has a code. And what happens is every 10 there's a bridge, and then every 5 there's a bridge, and then every 6 there's a bridge, and then every 5 there's a bridge, and then it repeats over and over and over and over again. And that's a part of your DNA, that's a part of my DNA. This is something that we just recently discovered. This whole thing of 10, 5, 6, 5. Well, what is amazing is with the numbers actually correspond with Jewish letters. The number, if you notice that there's 2 number 5s, both of those are H's. And then if you'll notice that the number 10 is a Y and the number 6 is a W, it's how the Jews spell Yahweh. And God's name is literally written in your DNA.
Now, I'm gonna be honest, I don't think this room has a very academic vibe to it, so we're gonna go somewhere a bit more appropriate so we can discuss the structure of DNA. Now, DNA is a polymer. It's a polymer found within each and every one of our cells, and polymers are basically large organic molecules made up of numerous smaller molecules known as monomers. The monomers are basically like the building blocks or subunits that make up the overall polymer. All monomers within a polymer will have effectively the same structure, or at the very least, very similar structures. To give an analogy, you can think of a polymer almost like a large Lego structure, say like a tower or something like that. And the individual building blocks, the individual Lego blocks that are used to make up that tower would be the monomers. Now, most of you are probably already familiar with the double helix structure of DNA. If that term's unfamiliar to you, it's basically just the overall shape of DNA, this weird wiggly ladder-like structure. Now, this double helix is basically the polymer. However, this polymer is made up of numerous monomers within it. Each of these monomers are known as nucleotides. So, the nucleotides are basically the monomers or the basic building blocks that make up the polymer that is DNA. However, the nucleotides, the building blocks of DNA, are in and of themselves also made up of building blocks. Nucleotides have 3 main components to them. They have a sugar, which I'm just going to be representing with this hexagon structure. They have an attached phosphate group, and last but certainly not least, they have a nitrogenous base. The nitrogenous base, this structure right here, this is what mainly distinguishes one DNA nucleotide from another. Basically, every single DNA nucleotide is identical when it comes to the phosphate group and the sugar group. Every single one of the DNA nucleotides will have the exact same phosphate and sugar group. However, they differ with regards to what their nitrogenous base is, and that's how you can distinguish one nucleotide from another. In fact, there are 4 different nitrogenous bases that DNA can have. The 4 nitrogenous bases found in DNA are known as adenine, guanine, cytosine, and thymine, otherwise known as A, G, C, and T for short. Now, I'm just going to quickly draw a diagram of all 4 of the different nitrogenous bases so that you can kind of see the difference between all 4 of them. Now, as you might have been able to notice, C and T, or cytosine and thymine, both have very, very similar structures, and guanine and adenine, or G and A, both have very similar structures, with both A and G containing 2 different rings in its structure, whereas C and T only contains a singular ring in its structure. These 2 structural differences are actually very notable because it allows us to classify the nitrogenous bases. For example, the A and G bases are referred to as purines, whereas the C and T bases are referred to as pyrimidines. I should also note that I tried to color code the diagrams a bit. The oxygen atoms are labeled in red, whereas nitrogen atoms are labeled in green. Any carbon or hydrogen atoms are being labeled in white, and basically every single one of these little intersections between 2 lines, that qualifies as a carbon atom. And these double lines are just to represent a double bond between 2 carbon atoms. Now, while most of you are probably already familiar with DNA, many of you may also be aware of a very similar molecule in our cells known as RNA. It has a very similar structure to DNA, mainly because it's made of nucleotides in the same way that DNA is. And these nucleotides for RNA also consist of a sugar, a phosphate group, and a nitrogenous base. Now, there are, of course, differences in the structure of RNA compared to DNA. And in fact, when we look at the individual nucleotides that make up RNA and DNA, we'll notice that there's actually two major differences between RNA and DNA nucleotides. And one of those differences actually has to do with the nitrogenous bases themselves. DNA has the following nitrogenous bases in it. However, RNA does not actually share all of these nitrogenous bases with DNA. RNA, it does actually contain adenine, guanine, and cytosine. However, RNA does not contain thymine. Instead, they have a different nitrogenous base in place of thymine, known as uracil. I'm going to quickly draw the structure of uracil so that you can compare it with thymine. What you'll notice about the structures of thymine compared to uracil is that, for all intents and purposes, they're almost identical. There is only one difference between the structure of thymine compared to the structure of uracil, and that is this carbon atom right here. In uracil, this carbon atom is basically only attached to a hydrogen atom over here. However, in thymine, this carbon atom is actually attached to what's known as a methyl group, and a methyl group is basically a carbon with 3 hydrogen atoms attached to it. A methyl group is almost like methane. Methane is basically CH4, so a carbon with 4 hydrogens attached to it, and a methyl group is basically when you have methane, except we scrap one of the hydrogens and then attach that methyl group to another carbon or some other atom of some sort. So in short, when we look at RNA compared to DNA, what we notice is that with the nitrogenous bases, DNA will contain the nitrogenous bases A, G, C, and T, whereas RNA will contain the nitrogenous bases A, G, C, and U. And the difference between the thymine or T and the uracil, i.e. U, is very minimal. It doesn't seem like much of a difference, but the difference is still significant enough that RNA can be easily distinguished from DNA just by using the nitrogenous bases. On a bit of a side tangent, earlier I mentioned that adenine and guanine are both purines and that cytosine and thymine are both pyrimidines. I should also note that uracil is a pyrimidine as well. Now, purines are pure as gold. However, for the pyrimidines, cytosine, thymine, and uracil are all pyrimidines. Now, when I think of the term pyrimidine, the first thing that comes to mind are pyramids. So, in order to remember which nucleotides are the pyrimidines, you can think of the following saying. You need to cut stones in order to make a pyramid. So, you can remember which nucleotides are purines and which are pyrimidines by remembering that purines are pure as gold, and that when it comes to pyrimidines, you need to cut stones from the pyramid. Now, as mentioned earlier, nitrogenous bases only make up 1/3 of the structure of an individual nucleotide. So we're going to move on from nitrogenous bases and move over to the sugar that makes up the nucleotides of DNA and RNA. You might be aware that DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. Now you might notice those two terms sound very, very similar, and the reason for this similarity has to do with the structure of the nucleotides of DNA compared to RNA. The biggest difference between DNA and RNA in terms of its structure is that DNA is made of deoxyribonucleotides, whereas RNA is made of ribonucleotides. The difference in these two terms is the hint we need to figure out what differentiates deoxyribonucleotides from ribonucleotides. And this hint is in that term, deoxy. This signals that DNA nucleotides lack an oxygen atom where the normal ribonucleotides of RNA would normally have an oxygen atom. And this oxygen atom that's found in RNA but isn't found in DNA is located on what's known as the 2' carbon of the sugar of RNA. Said oxygen atom is absent on the 2' carbon in deoxyribonucleotides. Now, that sounds like a very small difference, yet much like how uracil differed from thymine with the nitrogenous bases, that small difference is significant enough to differentiate the structure of DNA from RNA completely. They have completely different structures and completely different functions in our bodies as a result. Okay, so I took the liberty to draw the structures of deoxyribose and ribose, which are basically the sugars that are found in deoxyribonucleotides and ribonucleotides respectively. And what you'll notice is that when you're looking at deoxyribose compared to ribose, there is basically no difference between the two. However, there is, of course, an oxygen atom that is found in ribose on the 2' carbon located here. And in deoxyribose, this oxygen is absent. And it's in the exact same location in terms of which specific carbon of the sugar contains or lacks that oxygen. So just by looking at the structures here, you can kind of see how deoxyribose and ribose are very similar, and the only difference is that deoxyribose lacks an oxygen where ribose normally has one. Lastly, but certainly not least, let's talk about phosphate groups, the third and final component that makes up the nucleotides of both RNA and DNA. Both RNA and DNA can have anywhere from 0 to 3 phosphate groups attached to them on the 5' carbon of their sugar. Brief review. So, in nucleotides, both RNA and DNA nucleotides have the sugar, a nitrogenous base, and anywhere from like 0 to 3 phosphates. Now, these phosphates are very important, except when it comes to differentiating DNA from RNA. DNA and RNA don't actually differ with regards to their phosphate group. They actually have basically identical phosphates. The phosphate groups, however, are very important when it comes to the overall structure of the polymer that is DNA, because it is these phosphates that actually allow you to go from individual nucleotides to the polymer that is DNA or RNA. So if it wasn't for these phosphate groups, you would only have the individual nucleotides. But because of these phosphate groups, we can form bonds that allow us to form the overall polymer that is DNA. The bonds that are formed between nucleotides using the phosphate groups are what are known as phosphodiester bonds. Basically, what we have is that the phosphates of the 5' carbon located here, please don't mistake this for the phosphates, I'm just circling this carbon. Basically, the phosphates that are attached to the 5' carbon will bond with the 3' carbon on the sugar of the next nucleotide. This will basically link the two different nucleotides together. And after millions upon millions of these links occur, you then get the full-fledged strand of DNA. I'm going to try to do a very rough diagram to show what I mean. You see here that the phosphate group attached to the 5' carbon of this first nucleotide is also attached to the 3' carbon of the next nucleotide. And similarly, in this second nucleotide, the phosphate group attached to its 5' carbon is then subsequently attached to the 3' carbon of the next nucleotide, and so on and so forth. We basically get a linkage between each and every one of the different nucleotides by linking the phosphate group, normally found on the 5' carbon, with the 3' carbon of the next nucleotide. Now, I'm probably going to also show you a better diagram of these phosphodiester bonds, because I do recognize that this is a little bit bad. But at the same time, just note that with these phosphodiester bonds, you're able to link one nucleotide to another, which eventually allows us to form the overall polymer of DNA or RNA. So both DNA and RNA are linked via these phosphodiester bonds. Now, I just drew a very, very rough diagram of DNA and RNA respectively, and you might notice that DNA is typically double-stranded within a cell, whereas RNA is more often than not single-stranded in the cell. Now, both DNA and RNA are linked via phosphodiester bonds. The individual nucleotides in RNA or in DNA are linked together via these phosphodiester bonds. But you might notice that it is not these phosphodiester bonds that cause DNA to be double-stranded, because if it was, we'd expect RNA to also be double-stranded. So, it is not actually these phosphodiester bonds that link the two strands of DNA. Something else must be linking these two strands together. So, what exactly is it? When we look at DNA, it has this double helix structure. And the outside section of the DNA, so like this section right here, this is what's known as the sugar phosphate backbone, mainly because it is composed of the sugar and phosphate groups. So basically, if I'm to draw DNA, and let's just say I didn't include the stuff in the middle here, and if we're only looking at this, this is the sugar phosphate backbone, meaning it's made up of the phosphate groups and the sugar groups. It is excluding these nitrogenous bases. Now, in the center, these, what I would basically deem as the rungs on the ladder of DNA, are the nitrogenous bases. Now, what's really notable about DNA is that it is perfectly complementary. You find that when we look at the two different strands of DNA, one strand is going to be perfectly complementary to the other strand that it's paired up with. Let me go into a little more detail on that. Now, you might remember earlier we talked about purines and pyrimidines. So the purines are pure as gold, and the pyrimidines are cut. Now, we're going to exclude the U in this instance since we're talking about DNA and not RNA. What we find is that the pyrimidines pair up with the purines. Basically, if we were to look at the two strands of DNA and say on the left strand we have an A base, followed by a G base, followed by C, and followed by T. What we find is that, so the nitrogenous bases of the pyrimidines always pair up with purines, and they do so in a very specific way. A always pairs up with T. G always pairs up with C, and vice versa. C always pairs up with G, and T always pairs up with A. And this is why we say that the two strands of DNA are complementary to each other. If you have one sequence of nucleotides like A, G, C, and T, the other DNA strand will be complementary to it, having the sequence of T, C, G, and A. Now, how exactly do these nucleotides pair up with each other? Well, they pair up with each other through the use of what are known as hydrogen bonds. So, it is through these hydrogen bonds that the two different strands of DNA are connected. I'm just going to represent the hydrogen bonds like this. So there are hydrogen bonds that connect the two different strands of DNA together. Now, DNA is often referred to as complementary, but it's also referred to as antiparallel. Well, what does that mean? Well, basically it means that if we were to look at the DNA from, let's say, left to right, what we would find is that one strand looks like it's running in one direction, while the other strand looks like it's running in the other direction. So let's just say, for instance, the top strand looks like it's running in the 5' to 3' direction, whereas the bottom strand is running in the 3' direction to 5' direction. And these are just the nitrogenous bases in between the two strands. Now, the antiparallel nature of DNA may sound confusing at first, but you might be able to understand it using an analogy. Let's just say for a moment, you are on a road. You're driving on a road, and you notice it's divided by a yellow line. What that indicates is that the cars on this side of the line will be going in this direction. They'll be going upwards, whereas the cars on the left side of the line will be going downwards. And so we also have a very similar thing with DNA. On the one strand of DNA, it's kind of going in the 5' to 3 direction rightwards, whereas on the other strand of DNA, it's going in the 5' to 3' direction leftwards. So that's how you can understand the antiparallel nature of DNA. So now we know that DNA is both antiparallel and complementary. But let's just go into a little more detail to show what I mean by that. Let's just say we have one DNA strand, and the sequence of that DNA strand is this. So on one strand of DNA, this is what we have. What we would expect to see, therefore, is that the opposite strand of DNA would look something like this. The little lines I drew in between the letters are basically just being used to represent the hydrogen bonds in between the two strands of DNA. So what we see here is that because the DNA strands are antiparallel, it means that the top strand is running in the 5' to 3' direction, whereas the bottom strand is running kind of 3' to 5' direction, at least if we're looking left to right. And because DNA is complementary, we notice that the top sequence is perfectly complemented by the bottom. Every single A in the top sequence has a T nitrogenous base complementing it. Every single G has a C base as a complement, every single C has a G base as a complement, and every single T has an A base as a complement. Thus, we see that the two strands of DNA are antiparallel, but also perfectly complementary. All right, so now we should very clearly understand why we say that DNA is antiparallel and complementary. Now, I mentioned earlier that A pairs with T and C pairs with G because of what are known as hydrogen bonds. But the thing is, I never really actually went into detail about what hydrogen bonds are. And it seems kind of meaningless for me to say, oh yeah, A pairs with T and C pairs with G as a result of hydrogen bonds if I don't actually tell you what those bonds are. So, what exactly are hydrogen bonds? Well, hydrogen bonds are a form of intermolecular force, or IMF for short. Intermolecular forces are basically attractive forces that take place between two different molecules. You can think of this and better understand it by just thinking about the term intermolecular force itself, because it's an intermolecular force. In the same way that an international event is an event that involves more than one nation, an intermolecular force is a force that takes place between more than one molecule. It's not just the forces that hold atoms together within a molecule, it's the forces that hold two different molecules together. This is a rough diagram of water. So the forces that hold the two water molecules together would qualify as an intermolecular force, whereas intramolecular forces would be those that hold the atoms within a molecule together. Okay, so hydrogen bonds are a type of intermolecular force, but how exactly do hydrogen bonds form? Well, to understand this, we need to understand a couple other elements. In particular, we need to understand fluorine, oxygen, and nitrogen. These are three elements you will find on the periodic table, and what's notable about these three elements in particular is that they're very strongly electronegative. And what this means is that they very strongly attract electrons towards themselves, more so than most other elements on the periodic table. So these guys really strongly attract electrons. They're like an electron vacuum compared to most other elements. Now, when fluorine, oxygen, or nitrogen pairs up with hydrogen, I'll just use oxygen for this example. When oxygen is paired with hydrogen, it forms what's known as a covalent bond, which is basically a bond in which the electrons are shared between oxygen and hydrogen. However, as mentioned before, oxygen is very electronegative, meaning that when they share electrons with each other, it's actually kind of unequally shared. The oxygen kind of hogs most of the electrons closer towards itself compared to hydrogen. The uneven sharing of electrons forms a dipole, and what this means is that basically oxygen becomes partially negatively charged because electrons are negatively charged, and hydrogen becomes partially positively charged because it's losing some of its electrons, or at the very least, it's having these electrons unequally shared with it. And this is what we mean by dipole. Basically, one end of the molecule is more negatively charged, while the other is more positively charged as a result of this unequal sharing of electrons. Now, it is this partial positive charge found on hydrogen that explains hydrogen bonds. Basically, we see that when we have this sort of... Here, I'm going to draw another molecule. Let's draw a molecule of water. This red circle in the center is just being used to represent oxygen, and the two hydrogens on the side are colored in white here. So let's just say we have two different molecules of oxygen in a nearby proximity. As mentioned before with water, because the very strongly electronegative oxygen is covalently bonded to the less electronegative hydrogen, it means there will be an unequal sharing of electrons, making this oxygen partially negative, while the hydrogen is partially positive. When it comes into contact with another water molecule, its oxygen is largely negative. And because hydrogen is partially positive, Well, positive attracts negative. Think of a magnet, for example. The positive end of a magnet will be strongly attracted to the negative end of a magnet. And the same thing goes here. The positively charged hydrogen atoms on this water molecule will be strongly attracted to the negatively charged oxygen atom on this water molecule. And this attraction results in a strong bond between the two, referred to as a hydrogen bond. Similarly, in this other water molecule, the partially positive hydrogen of this water molecule will be strongly attracted to the partially negative oxygen of the adjacent water molecule, which results in a hydrogen bond between the hydrogen of this water molecule and the oxygen of this water molecule. And this is basically what a hydrogen bond is. It's just a strong attractive force between the partially positive hydrogen atoms of one molecule to the negatively charged atoms of an adjacent molecule. I use water in this example mainly because water is one of the molecules where we very, very commonly see hydrogen bonds. And in fact, hydrogen bonds explain some of the very weird traits of water. One really good example is the surface tension of water. You might notice that water's surface tension is actually pretty strong. In fact, if you've ever had the misfortune of being in a public outdoor pool, you will have probably noticed that many bugs and spiders might actually have been crawling on the surface of the water. And that's because in order to break the surface tension of the water, you effectively need to break several of these hydrogen bonds. And hydrogen bonds are actually pretty tough to break compared to a lot of intermolecular forces. So you might find that a lot of bugs are able to walk on the surface of the water, but you drop something heavier and with enough force, it'll very quickly go under the surface of the water. And that's just because, well, it has the force to actually break some of these hydrogen bonds. Now, I know I've used the example of water for much of these hydrogen bonds, but I should note, whenever hydrogen is bonded to a more electronegative atom like O, F, or N, this gives the hydrogen atom a partial positive charge that enables hydrogen bonding with an adjacent molecule that has some sort of electronegative atom on it. For example, the hydrogen of this molecule could bond to a fluorine, oxygen, or nitrogen atom of an adjacent molecule via a hydrogen bond. So hydrogen bonds aren't just reliant on, like water. It most commonly occurs in water, but it can occur between any sort of molecule where there's this NH, FH, or OH bond, and if there happens to be an adjacent FO or N atom in a nearby molecule. I should note, though, hydrogen bonds are one of the strongest intermolecular force you can get. So it is a very strong attractive force. However, hydrogen bonds are still weaker than the forces that hold atoms together within a molecule. For example, the covalent bond between nitrogen and hydrogen here, or between oxygen, hydrogen, or fluorine and hydrogen here, these covalent bonds between atoms within a molecule are significantly stronger than the hydrogen bonds that might form between hydrogen and an adjacent molecule. And I do want to emphasize, hydrogen bonds are not bonds between the atoms within a molecule. They are not the same thing as the bonds that hold the atoms within a molecule together. So the bonds that basically make a molecule are intramolecular forces. Hydrogen bonds are not intramolecular forces, and rather they take place between two different molecules. Okay, sorry for that tangent, but hopefully now you can understand better what a hydrogen bond is. And with that in mind, we can now better discuss how the two strands of DNA actually connect with one another. As mentioned before, the two DNA strands are connected via hydrogen bonds between the nitrogenous bases. Beforehand, we were drawing the DNA kind of like this, where the sugar phosphate backbone of the DNA is shown here and here, and the nitrogenous bases are shown here. Now, when we draw the nitrogenous bases this way, it almost makes it look as though it's like one continuous thing. But now that we know a bit more about hydrogen bonds, it might be more accurate to draw the DNA kind of like this. This way, we're showing that the nitrogenous bases of DNA are not like fused with the nitrogenous bases on the other strand of the DNA. Rather, there are hydrogen bonds that form between the nitrogenous bases of the two opposite strands. Now, while I mentioned that hydrogen bonds connect the two strands of DNA, one thing I forgot to mention is that the different nitrogenous bases actually have different numbers of hydrogen bonds. For example, When C is paired up with G, it actually forms 3 separate hydrogen bonds between the 2 different nitrogenous bases. Similarly, when A is paired with T, we actually get 2 hydrogen bonds that form between the nitrogenous bases of A and T. So adenine and thymine only form 2 hydrogen bonds when paired up, whereas cytosine and guanine form 3 hydrogen bonds when they're paired up. Now, I'm probably going to try to put an image on screen right now to show the exact molecular diagram that illustrates these hydrogen bonds. I'm not going to draw that molecular diagram myself, mainly because I think it would be a little bit too complicated. It would probably take up too much time, and chances are I'd probably draw it poorly anyway.
All right, so today we're going to be considering a sequence in an arbitrary metric space, and we're going to be talking about...
What are you doing? What are you doing?
A bow mass video.
Okay, I can see that.
Yeah.
I left my camera here for 5 minutes, and you decided that you would just use this for the video? There's a reason I told you you're not... There is a reason I told you are not allowed on my channel again. Okay, get out, get out. Sorry about that, guys. I went to go get a drink of water. I was gone for 5 minutes, and next thing I know, that Bowmaths guy is filming one of his new math videos using my camera. The audacity of some people, am I right? Make sure not to check out that guy's channel, by the way. Do not, under any circumstances, go check out the channel Bowmaths, linked in the description below and on screen now. The dude's a total nerd. All he does is sit around and make math videos all day. He's just doing math all day like a nerd. And as you can clearly see, we don't allow nerds on this channel. This is a nerd-free zone. All right, so with all of this in mind, let's zoom out and look at the big picture of DNA, knowing all that we do now. Now, on a macro scale, when we look at DNA, it's often represented as this almost spiraling ladder-like structure. The outside parts of the ladder are the sugar phosphate backbone of the DNA, so-called because it consists of the sugar and phosphate components of each of the DNA nucleotides. In the middle, the rungs of the ladder are basically the nitrogenous bases of the DNA. And these nitrogenous bases of the DNA are held together through the use of hydrogen bonds between the nitrogenous bases, with A and T having 2 hydrogen bonds between each other, and C and G having 3 hydrogen bonds between each other. Now, in addition to that, we see that DNA is complementary. For every single A on one strand, there is a complementary T on the other strand. For every single C on one of these strands, there's a complementary G on the other strand just opposite to it. Similarly, for every T on strand 1, there's an opposite A on strand 2. For every G on strand 1, there is an opposite C on strand 2 just across from it. However, DNA is also antiparallel, which means that the 2 different strands in DNA actually go in opposite directions. For example, the strand shown here on the right goes from the 5' end all the way down to the 3' end if we're going top to bottom. If we go top to bottom with the second strand, it goes from the 3' end all the way down to the 5' end. Now, you might have noticed I keep referring to the 5' end and the 3' end. And the thing is, I forgot to define what those actually mean. What is the 5' end? What is the 3' end? Well, when we look at the structure of DNA, recall that the nucleotides of DNA are linked together via phosphodiester bonds. Basically, the phosphate groups of one nucleotide link up to the 3' carbon on the sugar of the next nucleotide. So, when it comes to connecting the nucleotides in a single strand of the DNA, we see that the phosphates link up with the 3' sugar of the next nucleotide. And this keeps happening over and over until you get to the very last nucleotide. Now, the 5' end is the end in which the 5' carbon isn't linked to another nucleotide. The 3' end is where the 3' carbon isn't linked to another nucleotide. Recall that this carbon on the sugar shown right here, that is the 3' carbon. Similarly, that carbon located right there, which would effectively be right there on the diagram, that is the 5' carbon of the sugar. Now we see that on the 5' carbon of the sugar, we have the phosphate groups attached. And on the 5' end, we notice that unlike all the phosphate groups here, the phosphates on the 5' end of the DNA are not attached to another nucleotide. And that's how you can recall that this is the 5' end. Similarly, when we look at the 3' end of the DNA, we see that on the 3' carbon, instead of that carbon being attached to a phosphate group like all these other nucleotides, here on the 3' end, the 3' carbon is attached to a hydroxyl group, otherwise known as an OH group. So we don't have another nucleotide being linked to this last one on its 3' carbon. And what that means is that the 3' end is the end of the DNA in which there aren't any nucleotides attached to the 3' carbon. And that helps you understand why DNA has directionality. It has a direction because we have a distinct 5' and 3' end of the DNA. And if DNA has directionality in terms of the 5' and 3' ends, that helps us understand how DNA can be antiparallel, because the two strands of DNA run in opposite directions. Now that we know about the structure of DNA in significantly more detail, let's go back and rewatch that original video, because in rewatching it, we might be able to now notice exactly what's wrong with it, given what we've learned about the structure of DNA.
In your genetic code, it's a double helix that right turns, and there are these sulfur bridges in the midst of every single atom that you have that has a code. And what happens is every 10, there's a bridge, and then every 5, there's a bridge, and then every 6, there's a bridge, and then every 5, there's a bridge, and then it repeats over and over and over and over again. And that's a part of your DNA, that's a part of my DNA. This is something that we just recently discovered, this whole thing of 10, 5, 6, 5. Well, what is amazing is with the numbers actually correspond with Jewish letters. The number, if you notice that there's two number 5s, both of those are H's. And then if you'll notice that the number 10 is a Y and the number 6 is a W, it's how the Jews spell Yahweh. And God's name is literally written in your DNA.
So, rewatching the video, there are a lot of flaws with it. For one, he states that in the midst of every atom, there are sulfur bridges. Now, I'm assuming that this is him misspeaking. After all, you can't exactly have sulfur bridges between each atom, unless, of course, every single atom in DNA is a sulfur atom. Secondly, he claims that these sulfur bridges have a code to them. Once again, I'm willing to chalk this up to him maybe misspeaking or misunderstanding something. Because when we think of the DNA code does not come from these sulfur bridges. Rather, when someone is talking about the genetic code, what they mean is the sequence of nucleotides. For example, here, the code of the DNA consists of the order of these nucleotides shown here. The third big issue that I have with this video is that he simply states that, quote-unquote, every 10 there's a bridge, then every 5 there's a bridge, then every 6 there's a bridge, then every 5 there's a bridge. And this forms a repeating 10-5-6-5 pattern of sulfur bridges in the DNA. My biggest issue when he states this is that he doesn't specify what he actually means when he states that every 10 there's a bridge, every 5 there's a bridge, every 6, every 5 there's a bridge. My best guess is that he's referring to the numbers of nucleotides. So what he's saying is that, let's say we start with a sulfur bridge right over here. What we would then do is then we'd count about 10 nucleotides, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then there's a bridge. Then another 5 nucleotides, 1, 2, 3, 4, 5, there's a bridge. Then 6 nucleotides, 1, 2, 3, 4, 5, 6, there's a bridge. Then another 5 nucleotides, and we get a bridge. And so then we get this repeating pattern of sulfur bridges, where you get a sulfur bridge followed by 10 nucleotides, then a sulfur bridge followed by 5 nucleotides, then a sulfur bridge followed by 6 nucleotides, and last but certainly not least, a sulfur bridge followed by 5 more nucleotides. So, I will admit, as it stands, I'm already being very critical of the video, because he seems to have misspoken at least twice in the video, and when it came to the main claim, the most critical point of the video, he's very unclear about what he actually meant by it. This is my best interpretation. Now, you might be wondering, aside from these nitpicks, is there anything that's actually fundamentally wrong with the video, such that it kind of invalidates the claim that is being made in it? And the answer to that question is yes. One of the biggest issues with the claim he makes is that he claims that there are sulfur bridges that hold DNA together. We do notice that in proteins, the disulfide bridges are used to hold the amino acid chain together in a certain way. And this is very important, because the way in which the amino acid chain is folded impacts the structure of the protein. and the structure of the protein impacts its function. You can think of the structure of protein impacting its function kind of like tools. The structure of a tool helps to determine its function. A hammer, for example, has a very big blunt end on it for hammering a nail, whereas a screwdriver has a very long, thin implement in order to screw screws into. Similarly, with proteins, we find that depending on the structure of the protein, it will have a completely different function. And if you change the structure of the protein, this can often change, or in the worst case scenario, inhibit the protein from functioning. That's a bit of a side tangent, I apologize. What's notable is that in the video, he claims that there are sulfur bridges that join the two strands of DNA together. And this is the biggest issue with the video. The two DNA strands are not held together by disulfide bridges. Rather, as we discussed earlier, the two strands of DNA are held together via hydrogen bonds between the nitrogenous bases of the DNA. So, one of the main reasons this video is wrong is because it claims that sulfur bridges, as opposed to hydrogen bonds, are what hold the DNA together. And in fact, what's most notable about this video is that there's no sulfur in DNA at all. There is not a single sulfur atom in DNA. In fact, we should be very glad that DNA doesn't have any sulfur in it, because if it did have sulfur in it, we may have never found out that DNA is actually the material that stores and passes down our genetic or hereditary information. Why do I say this? Basically, a long time ago, in the distant past of 1952, biologists were basically having a big debate. They knew that parents would pass down specific traits to their offspring. The only issue is that, well, biologists didn't exactly know how that was happening. They had two good candidates, though. Basically, biologists were debating whether DNA or proteins were the molecule through which we basically inherited traits from our parents. Is it that we inherited traits from our parents because they passed on their DNA to us? Or did they pass on their proteins to us, and it was through the proteins that we inherited specific traits that our parents had? This was the big debate back in the 1950s, and two researchers named Hershey and Chase wanted to determine which of these two molecules was the cause of inheritance. In order to determine this, they wanted to do a specific experiment, and this experiment would use bacteriophages. Now, as the name kind of implies, bacteriophages are a type of virus, but they're specifically a virus that infects bacteria. This is kind of the structure of a bacteriophage. Now, during the 1950s, we did know that bacteriophages consisted of two components. They had an outer protein shell, so this big structure of the virus is made out of proteins, and within what's referred to as the head of the bacteriophage, you have DNA. Now, another thing that we knew about bacteriophages at the time is that bacteriophages reproduce by infecting bacteria. Basically, what happens is that the bacteriophage latches onto the bacteria and injects something into the bacteria. So it injects a specific substance into the bacteria. What this substance is wasn't exactly known at the time. All we knew is that this substance that was injected into the bacteria carried the hereditary information of the bacteriophage. And we knew this because once this substance was injected into the bacteria would effectively become a factory for producing these viruses. It would take this hereditary information from the bacteriophage and basically produce a bunch of viruses. So what we knew at the time when Hershey and Chase were doing their experiment is that the bacteriophages are made of two components, a protein component and a DNA component. And what we also knew is that these bacteriophages, when they infect bacteria, they insert something into the bacteria that carries their hereditary information. So, what was this mystery substance that they were injecting into bacteria? In order to determine what the hereditary substance that the viruses were injecting into the bacteria was, Hershey and Chase basically did two almost identical experiments. In one experiment, shown on the top here, they took several bacteriophages and used them to infect bacteria that were labeled with radioactive sulfur. They labeled several amino acids with radioactive sulfur. when a virus infects a bacteria cell, it takes all the resources and the machinery of the bacteria and uses them to turn the bacteria cell into a virus factory. It uses that bacteria cell to then mass produce more viruses. What this means is that if the bacteria have radioactive sulfur in their amino acids, it means that when the viruses infect the bacteria, that radioactive sulfur will be incorporated into the protein shell of the bacteriophages that come about from this infection. So, after infecting these radioactive sulfur-labeled cells of the bacteriophages, the resulting phages would have a radioactive protein shell. I've labeled the radioactive protein shell in green. Basically, whenever there's radioactive sulfur in this diagram, I've tried to label it with green. Now, a very similar test was done in order to determine whether or not DNA was the material through which inheritance came about. Hershey and Chase used bacteriophages and infected several cells that were labeled with radioactive phosphorus. Now, the reason they did this is because phosphorus is found in DNA, but it is not found in proteins. So, when the virus infects these cells, it basically takes up the radioactively labeled nucleotides and incorporates it into its own DNA. And because we could better visualize these radioactive elements, we now had two different groups of bacteriophages. One group of bacteriophages had a radioactive protein shell, while the other group of bacteriophages had a radioactive DNA. Now, what they did is then they took these two different radioactive viruses and used them to infect new cells. So, in both cases, what ends up happening is that the bacteriophages injected their DNA into the cells. However, because Hershey and Chase wanted to figure out what the bacteriophages were injecting into the cells, because they didn't know at the time whether or not DNA or protein was being injected, they then took these cells that were infected by the bacteriophages and literally put them in a blender. And the reason they put them in a blender is because by doing so, it would dislodge the bacteriophages from the cells. By dislodging the bacteriophages from the cells, it means that there wouldn't be any bacteriophages on the cells, but what would be left is whatever the bacteriophages injected into the cells. In this case, we know it was DNA, but at the time, like I said, they didn't know this. But by dislodging the bacteriophages from the cells, we could then determine what exactly was being injected into these cells. After the viruses were dislodged from the cells using blenders, the sample in which the viruses and cells were contained in was then centrifuged. Basically, they put it in a machine that spun the sample around extremely quickly. And by spinning the sample like this, it would allow the heavier stuff in the sample, i.e. the cells, to sink to the bottom of the tube, whereas the lighter viruses would be able to basically be in the top, more liquid portion of the tube. After centrifuging both of these samples, what they found is that in the sample in which they basically used radioactive sulfur to label the proteins, they found that basically all of the radioactive sulfur was only found in the viral portion. None of the cells that were infected by these viruses seemed to have any radioactive sulfur in them. In contrast, for the cells that were infected with bacteriophages that were labeled with radioactive phosphorus, they found that the viruses didn't seem to have any radioactive phosphorus in them. In comparison, the cells that had the DNA of the viruses injected into them were heavily saturated in radioactive phosphorus. And this seemed to indicate that it was the DNA rather than the protein that was being injected into the cells. And this would indicate that it's actually the DNA that carries our genetic material. However, Hershey and Chase then confirmed this by allowing these cells to act as, well, virus factories. They allowed the cells to basically produce several viruses within them, and then they broke open the cells in order to release all the viruses. When they looked at the offspring viruses, the viruses that originally came from these viruses that had radioactive sulfur in them, they found that the resulting viruses were completely normal. There wasn't any radioactive sulfur in them, and this seemed to indicate that the radioactive sulfur was not passed down from the parent viruses to the offspring viruses. However, when they looked at the viral sample that injected radioactive phosphorus-labeled DNA into the cells, what they found is that the viruses that came from these original viruses still had radioactive phosphorus in their DNA. And what this indicated is that the offspring viruses inherited the radioactive DNA from their parents. And so, in short, what this experiment showed is that DNA is the molecule that carries the traits from parent to offspring. It is DNA that allows inheritance to take place. Now, you might be asking yourself, what does any of this have to do with the whole sulfur bonds being found in DNA claim? Well, what was really ingenious about this experiment is the fact that proteins do not contain any phosphorus in them, and DNA doesn't contain any sulfur in it, which means that when Hershey and Chase labeled the proteins using radioactive sulfur, there wasn't a risk of that radioactive sulfur also accidentally labeling the DNA. And vice versa. When they labeled the DNA with radioactive phosphorus, there wasn't a risk of that phosphorus also accidentally labeling the proteins. And that is why this experiment couldn't have actually been carried out if DNA had sulfur bonds in it. If DNA actually had sulfur in it, what we would expect to see is that these cells would not only contain radioactive phosphorus, but they would also contain radioactive sulfur in them. And this would make it extremely difficult to figure out whether or not DNA or proteins were the molecule of inheritance. Because for all we know, if DNA had sulfur in it, this result could indicate that either DNA is the molecule through which inheritance takes place, or alternatively, perhaps inheritance can only take place when both DNA and proteins are present in a sample. And thus, it would be a combination of DNA and proteins that allow for us to inherit traits from our parents. But because there isn't any sulfur in DNA, this experiment was able to work almost perfectly. So in short, if DNA actually contained sulfur in it, we may have never figured out that DNA is the molecule through which we inherit traits from our parents. And that is one of the reasons it's so important that DNA doesn't have sulfur in it. So, in order to make an extremely long story short, the biggest flaw that this video makes is the fact that they claim DNA is held together by disulfide bridges, or sulfur bonds. This is simply false. DNA has no sulfur in it to allow for these sorts of disulfide bridges to form. And if there's no disulfide bridges in the DNA, then there can't be a repeating 10-5-6-5 pattern that can form using these disulfide bridges. Now, normally for most people, this would be where the video ends. Okay, we found out that DNA has no sulfur in it, which means it's not possible for it to have any disulfide bridges, and what that means is that the claims made in this video are obviously false. Video debunked, it's all over, go home. But the thing is, I don't really want to end the video there, because when I first saw this video, I thought to myself, this couldn't just all be made up, right? I really do like giving people the benefit of the doubt, and in this video, the man in question making these claims said something that really made me think he might have confused sulfur bridges with something else in DNA. In particular, the guy in the video claimed that DNA right turns. And this phrase stuck out to me, because he's actually right about that. The DNA in our body has what's known as handedness. And in particular, DNA in our bodies is right-handed. But what does it mean for DNA to be right-handed? Well, when I say that DNA is right-handed, I mean it in the sense that the way in which DNA spirals, the direction in which DNA spirals, is considered right-handed. All right, so I just finished making a couple diagrams to illustrate the handedness of DNA. Now, as I mentioned earlier, the guy in the video claimed that DNA right turns. And if he is saying that DNA is right-handed, then he would indeed be correct. All of the DNA in our bodies is indeed right-handed, as illustrated here. There is such a thing as left-handed DNA, but it is extremely rare to find. You're not going to find any left-handed DNA in your own body. In fact, the conditions in which you find left-handed DNA are so rare, I can't even recall when you can find it. Just know it can't be found in the human body. So what exactly is the difference between left-handed and right-handed DNA? Well, to make a long story short, if you were to look at right-handed DNA from the top down, you would notice that both of the strands of DNA are spiraling in a counterclockwise direction. Now, when I was taking a biochemistry class, they illustrated the idea of right-handed and left-handed DNA in this way. If you want to consider right-handed DNA as like a screw, and you want to screw this piece of DNA into a piece of wood, how would you do it? Well, you would take your screwdriver and you would turn it in a clockwise direction. Because if we look at the top down, the DNA is spiraling in a counterclockwise direction, meaning to screw it downwards, we would have to be screwing it clockwise. I hope that didn't cause more confusion, because I do recognize that the handedness of DNA is sometimes a complicated topic, and if I do cause more confusion, I do apologize on that. So that's right-handed DNA. With left-handed DNA, what we find is that if we were to look at the left-handed DNA from the top down, we would notice that both of the strands of the DNA are turning in a clockwise direction, which means that if we wanted to screw this piece of DNA into a piece of wood, using it as a metaphorical screw, we would have to take our screwdriver and turn it counterclockwise in order to drive it downwards, because normally it's going upwards in a clockwise direction. So that is the difference between left-handed DNA and right-handed DNA. And like I said, to my surprise, this video seemed to have gotten one thing very right about DNA that not a lot of people know, the fact that DNA is right-handed. And when I heard this, I thought to myself, one of the reasons we call DNA right-handed compared to left-handed is because of the orientation of the major and minor grooves of the DNA. Basically, if we were to look at DNA like head-on, like a side view of the DNA, what we would find is that the major groove is a groove in the DNA in which more nucleotides would be exposed to us. So when we look at the DNA, our field of vision would have more nucleotides in it if it's in the major groove, and the minor groove would show fewer nucleotides. So I thought, well, maybe there might be a repeating 10-5-6-5 pattern in the number of nucleotides that are exposed in the different grooves of DNA. So I decided to try to look this up. And despite all the research I did, I couldn't find anything like this. The major groove seems to have roughly 5 or 6 nucleotides in it, whereas the minor groove had only about 3 nucleotides exposed to your field of vision. And this pattern just tended to repeat itself. So if anything, it wouldn't be like a 10-5-6-5 pattern. It would be more of a 5 or 6, 3, 5 or 6, 3, 5 or 6, 3. So there still isn't a 10-5-6-5 pattern that I could find in the DNA. So, as an effort to not be uncharitable, I tried my best to figure out whether or not this was just some sort of misinterpretation of an actual pattern that's found in DNA. And despite my best efforts, and despite all the research I put in, I couldn't seem to find anything related to this pattern of DNA. At the very least, I couldn't seem to find anything that was really credible. There were, of course, multiple Christian and Jewish websites that claimed that this pattern existed, but they didn't cite anything to actually show where they got this information from. So I did what any reasonable person would do, and I reached out to others. In particular, I reached out to 3 people who all have PhDs in relevant biological fields related to DNA. And I asked them, is there any 10-5-6-5 pattern in the DNA? Because I thought, maybe there is such a pattern in the DNA, but people were mistaking it for sulfur bonds for some reason. Despite my efforts, all 3 of the people I contacted had no idea what I was talking about. Such a pattern just doesn't seem to exist in DNA. And, well, at that point I was kind of at a loss. I did want to give this guy the benefit of the doubt, but all of my ideas were coming up short. I couldn't seem to find any credible information on a repeating 10-5-6-5 pattern in the DNA. But then, when I was almost at the end of my rope, I finally did it. I found out where this guy got his information from. This man got his information from a video made by someone named Dr. Israel Rubinstein, who claims that in 1986 he was researching DNA at the Weizmann Institute of Science. I'm going to just quickly play the video for you right now so you can get a picture of what I'm talking about.
אוקטובר 86, כשאתה היית פתק בכותל, או לפני זה, אני חקרתי במכון ויצמן, בתור רופא, של חולדות, עכברים, קופים ובני אדם. 18,000 רקמות של כבד, כליות, שרירים, בדקתי רקמות, ובדקתי אותם במיקרוסקופ האלקטרוני. לידכם, ילדים, במיקרוסקופ האופטי, שאתה מסתכל ככה, מגדיל 10,000. המיקרוסקופ האלקטרוני מגדיל 1,000,000 פעם. אני שם רקמת כבד, חולדת, עכברים, תמיד אותו דבר, אצל כולם. 2 סלילים הליקואידליים שהם משתלבים, זה נקרא DNA. ה-DNA נמצא גרעין, יש ציטופלזמה, הגרעין של הציטופלזמה, תאים. ציטופלזמה, בתוך הגרעין יש גופים שנקראים כרומוזומים. כרומוזומים, 22, 24, תלוי אם זה זכר או נקבה, הכרומוזום שבתוך הגרעין, אני לוקח את הכרומוזום, שם אותו ומראה לי את זה. זה היחידה של כרומוזום. מיקרוסקופית זה לא ככה, זה מחשב גדול, ענק, טלוויזיה, ואתה רואה, מה שהוא רואה מראה לך, זה אתה. סבבה. מדע, לא דת. ויש תוכנה במחשב שמדבר לי, שאלה שנייה. מה קורה פיזיולוגית? כשהשסלי לא יתפוצץ, לא ייפתח, הוא אומר לי שמדי פעם, sometimes, יש גשר של גופרית, שעושה שהסלים לא ייפתח. אני אומר, באמת, הגשר? גשר שמחזיק את הסלים, יפה מחזיק אותו. מחשב, מתי מופיע הגשר? אז מחשב אומר לי שיש חומצות נוקלאידיות על הבסיס של הסלים, דימין, סיטוסין, גואנינים, ATCG, שזה החומצות הנוקלאידיות של הסלים, והגשר של גופרית, סולפורו, מופיע ככה. כל 10 חומצות גשר, כל 5 חומצות גשר, כל 6 חומצות גשר, כל 5 חומצות גשר. ילדים, מה זה 10, 5, 6 חומצות? כמעט יש A, תף, A. תתי, בתוך ה-DNA, תתי, חתם את החתימה שלו, את השם שלו, בתאים שלך, בכל הגוף.
So as you saw in this video, he claims that he used a digital microscope to view the DNA. Now the thing is, I hadn't heard of a digital microscope before, but given the claims that he makes about this microscope, that quote-unquote optic microscopes enlarge images to up to 10 times their size, whereas a digital microscope enlarges the image to about 1 million times its size, I thought he might be referring to electron microscopes, and that's what he meant by digital microscopes. However, the way he described the microscope in the video was kind of bizarre, to put it lightly. The way he claims that he was able to communicate with the software. He claims that when he was viewing the DNA under the electron microscope, he was able to see several sulfur bridges that hold the two strands of DNA together. And I found this extremely strange, because he claims that he was able to discover these sulfur bonds as early as 1986. The thing is, if he did discover these sulfur bonds in 1986, this would have been a revolutionary discovery. This would have been taught in any biology and likely biochemistry class by this point. One of the reasons I know that this sort of information would be taught in schools worldwide is because we have had very similar discoveries made in biology just as recently as 1986 that have become widely known and widely discussed today. For example, as late as the 1980s, it was suggested that we classify organisms using what was known as the Five Kingdoms system. Basically, in this system, we classified all known living organisms into 5 separate kingdoms. These kingdoms included Monera, which basically includes bacteria and archaea, Protista, which includes protists, which by and large are mostly like unicellular organisms that aren't bacteria, but well, there's quite a lot of variety. They include basically most algae and stuff like that. Then there's also Plantae, which includes plants. The kingdom Fungi, which kind of self-explanatory, basically mushrooms, yeasts, stuff like that. And Animalia, which includes all living animals, like fish, birds, insects, humans. So this was the means by which we classified organisms for the longest time. These were the largest groups in which organisms were classified into. However, this all changed as late as 1990, mainly because it was discovered that many organisms we once considered bacteria were not actually bacteria at all, but they belonged to their own separate group. Certain organisms that we originally thought were bacteria because they, looked almost identical to bacteria, these not bacteria are referred to today as archaea. And shockingly enough, despite these archaea looking almost identical to bacteria, DNA analysis, along with a few other traits of archaea, show that they're actually more closely related to animals than they were to bacteria, and thus we had to change up the classification system. This led to the creation of an entirely new means of classifying organisms, known as the 3-domain classification. In the 3-domain classification, instead of grouping all organisms into 5 kingdoms, we group them into 3 domains: domain bacteria, domain archaea, and domain eukarya. The eukarya domain includes basically all of these kingdoms from the 5 kingdom system. It doesn't include Monera because Monera has now been split into the domains known as bacteria and archaea. Now, the reason I bring up this change to the classification system of organisms is because this change occurred in 1990. This discovery regarding archaea and their relation to eukaryotes occurred as late as 1990, and it's now widely taught today in biology classes. However, Dr. Rubenstein claims that his discoveries with DNA were found as early as 1986, and yet for some reason, nobody seems to know about them. If Dr. Rubenstein actually discovered these sulfur bridges in the DNA, why is it that no one seems to have any idea about them? It was then that I learned the unfortunate truth. As much as I wanted to give people the benefit of the doubt, he just made it up.
You really think someone would do that? Just go on the internet and tell lies?
Let's be real, we probably saw this from the beginning. He just made it up. He claims that he looked at the DNA under an electron microscope. The only big issue with this is that from what I can find, nobody had actually looked at DNA under electron microscopes until 2012. According to Dr. Rubenstein, he managed to observe DNA under an electron microscope 26 years before what we would consider the first instance of this taking place. And that seems a bit suspicious, to say the least, especially considering there seems to be absolutely no record of this incredible accomplishment that exists today. This, of course, raised my suspicions even further, and I kept digging and digging into Dr. Rubenstein's claims. And I learned that he's actually claimed these things multiple times. The only issue is that he did so in Hebrew. I do not speak Hebrew. I thought maybe that was where my search was going to end, but luckily for me, there were actually a lot of people who did speak Hebrew who have assessed and summarized some of Dr. Rubenstein's claims. And one of the biggest ones that really puts the nail in the coffin to this argument is this. Dr. Rubenstein claimed that he published his findings about the sulfur bonds in the DNA in October of 1986. And he claims that he published this data in the journal Nature. Now, people have purchased this issue of the journal Nature, and lo and behold, there is no such article written by Dr. Rubenstein in this journal. And thus, it seems that Dr. Rubenstein simply made this claim up out of thin air. There seems to be no actual articles, no actual evidence, no actual pieces of data that Rubenstein can point to in order to verify his claims, that God wrote his signature, wrote his name on our DNA. And in addition to that, no other researcher who has been working with DNA since 1986 has been able to find anything similar to what Dr. Rubenstein has claimed, which seems to indicate that, frankly, he just made it up. He made it up. He lied. So as much as I wanted to give people the benefit of the doubt on this one, it seems that the truth is shockingly simple. This man, Dr. Rubenstein, because of his religious convictions, thought it would be a good idea to make something up in order to bolster his religious truth claims. He simply lied in order to make his ideas seem more credible. And tragically, many Christians went along with this lie, assuming that it was true. And this is why we as Christians need to be very careful when it comes to presenting evidence and presenting arguments for the existence of God and for the truth of Christianity. If something sounds a bit too good to be true, it would be wise to check to see if it is too good to be true. And I want to make one thing clear with this video. I do believe that God has acted in the world and that we can actually see evidence for God. There is genuinely good evidence for the existence of God and for the truth of Christianity. In fact, at the very end of this video, I'm going to link 2 other videos that I've made which cover just some of the evidence for the existence of God and evidence for the resurrection of Jesus Christ. Because frankly, there is good evidence for Christianity. The only issue is that the argument I've just debunked ain't it. But with all that being said, thank you all so much for watching this video. If you enjoyed this video and, hey, if it helped you learn something new about DNA, I would highly appreciate it if you gave this video a like, subscribe to this channel, and hit the bell icon if you want to see more videos like this or any other theology videos I make. With all that being said though, may the grace of the Lord Jesus Christ and the love of God and the fellowship of the Holy Spirit be with you all. Go in peace.